Hey guys, so our next video is going to be on biotechnology. We're going to talk a little bit about recombinant DNA, and we're also going to talk a little bit about how biotechnology is involved in the field of forensics. So there's a little bit of vocabulary you need to be familiar with this unit. The first is what is recombinant DNA, and we're going to talk about this in a few more details in the next slide. But recombinant DNA is taking DNA from two different sources or organisms and combining it into one molecule. So recombinant DNA is a DNA molecule that contains part of the DNA from one organism or one source and the other part from another source. This is different from a transgenic organism, which is an actual organism that contains DNA from more than one organism. So don't get those two mixed up. And then we also talk a little bit about gene therapy, which is a really hot topic in biotech currently. And this is when scientists will replace a non-functioning or a damaged gene with a functioning gene. And this is usually to treat diseases. So I want to talk about the steps of making recombinant DNA. And this is important because this has a very modern use in that we use this technique of making recombinant DNA to produce insulin. So naturally your bodies produce insulin. However, people who are diabetic either do not produce insulin, or they do not produce enough of it to help regulate their blood sugar level. And so what scientists have done is developed a way to mass produce this insulin to then turn around and sell to people, especially people who are diabetic. And so to make recombinant DNA, there are 5 main steps. Step 1 is that whatever gene you want to produce, you need to isolate it and remove it. And you do that using something called a restriction enzyme, which we'll talk about in just a few moments. So if we're going back to the example of insulin, we're going to take that gene for insulin and remove it from an organism that naturally produces it. We're then going to take a DNA plasmid, which is a circular piece of DNA found in bacteria. The plasmid is going to be cut with the same restriction enzyme that we use to remove the gene. So the insulin gene that we're interested in, we cut that or removed it using a restriction enzyme. We're also going to cut the plasmid with the same restriction enzyme. We're going to combine those two, so we're going to take the gene that we want and our plasmid, put them together. We've now produced recombinant DNA, and then we're going to take that recombinant DNA and put it back into an organism. Again, if we're tying that back into insulin, that organism is going to be a bacterium. And we do that because bacteria can reproduce very rapidly. They do so exponentially, and it's very easy to maintain in a laboratory. So that bacterium is a transgenic organism which will undergo binary fission multiple times, and as a result will be producing insulin, which is what we want it to do. So we've now taken one gene and we have mass produced it in 24 to 48 hours, and that's how insulin is made. So I talked briefly about restriction enzymes, and here I'm going to focus a little bit more on what those restriction enzymes are. So remember, we've talked about enzymes. They are organic compounds, they're proteins that speed up chemical reactions. Well, here they're no different. Restriction enzymes are enzymes that are going to recognize specific short sequences of DNA and cut both strands of that DNA each time that sequence repeats. These sequences are palindromes, which means that they read the same forward as they do backwards. And so here's an example. If we look at the restriction enzyme EcoRI, EcoRI is going to recognize a 6 DNA sequence, GAATTC. Well, if you look on the 3' to 5' end, it's the same but backwards. Okay, so that EcoRI, it's going to read along the 5' to 3' end, and it's going to cut that DNA sequence anytime it sees GAATTC. And it's going to do so right in between the G and the A. Well, again, reading from 5' to 3' on the parallel strand, you can see it cuts right in between that G and A. So what it forms are these sticky ends. And so some enzymes produce sticky ends, some do something called blunt ends. So what we have is now restriction fragments. So here, if we're looking at this DNA sample, you will have 2 DNA fragments because that restriction enzyme cut the DNA at 2 spots. So restriction enzymes are super important. We're going to actually use them in a lab when we do a gel electrophoresis because it cuts that DNA up into those short sequences, which can then be spread across a gel. And we'll talk about that in just a moment. But one thing I want you guys to think about is that all humans have 99% of their DNA in common with one another. Only 1% is different, and that 1% is what makes each of us unique. So you should be able to conclude that if you use different restriction enzymes on different individuals, the restriction enzymes will cut that DNA at different spots. And what the importance of that is that some people will have longer fragments in certain areas, and some will have shorter fragments or restriction fragments. So this unique different sizes of restriction fragments per individual is what we call restriction fragment length polymorphisms. That's just a very fancy way of saying it's a very unique fingerprint for each individual. We all have different DNA, so the restriction enzymes will produce different sizes of those fragments. And this is important when you look at a crime scene. So let's say here, this DNA sample was obtained through a blood splatter at a crime scene. They have 3 suspects. They run their DNA using whatever or however they obtain that DNA, and they use restriction enzymes, but what you can see is that you have different banding patterns. So you're going to look for the one that matches up. If you look at suspect 2, it matches the sample at the crime scene. So this is really important in forensics and identifying criminals. So now we're going to switch gears. I've talked about a gel electrophoresis, and I want to go through the 4 main steps of that. You can either follow along on the diagram or use these words over here. So step 1 is to cut the DNA with the restriction enzyme. And I've just talked about what those restriction enzymes are, and there's several different ones. You're going to then take that DNA that was cut with restriction enzymes, and you're going to place them into wells in an agarose gel. You guys won't have to make the gels in class. It's a little bit old-fashioned, but I will talk about that in class, how that is typically made. The wells are these little loading areas at the very top of the gel. You're going to then run the gel, which means you're going to put it in a chamber, cover it with some buffer, and you're going to turn it on. And what it does is it's going to send an electrical current down that gel. So you have 2 ends, the cathode end, which is negative, and the anode end, which is positive. Remember, DNA is a negatively charged molecule because we have that phosphate attached to it. So you put the negative at the negative end, you turn on the power, and what will happen? Remember, opposites attract. So that negative at the top will move to the positive at the bottom. And because of those restriction enzymes, you're going to get different sizes of DNA moving down the gel. At the very end, you're going to take your gel and you would stain it with something called methylene blue. We've actually used that in a lab before, and it's going to make that DNA very visible. Now, one thing you guys should be able to conclude from this is that because we're separating DNA based on size differences, you're going to find your largest DNA at the top, or those really big restriction fragments at the top of your gel, and the really small ones at the bottom. And then that will give you that fingerprint or that DNA sample that you're looking for. We're going to wrap up this video very quickly by talking about a PCR, which is a polymerase chain reaction. And all you really need to know about this is that this is a process of taking a very small sample of DNA and amplifying it, or producing a lot of it. And to do a PCR, you need 3 things. You need free nucleotides, so A, G's, T's, and C's. You're going to need some primers, and you're going to need a Taq DNA polymerase, which is a special type of DNA polymerase that is used in this PCR. And PCR will do there's actually a machine. It'll do a series of heatings and coolings that will allow one single simple sample of DNA to become amplified and produce several of those. And that is a polymerase chain reaction.